Okay, thank you very much. Thank you for joining us today. We have an excellent presentation for you. Tiffany Burchill, one of our tech support specialists, is here to give you tips and tricks for ACD's miRNAscope assay. So with that, we'd like to get started. Thank you very much, Tiffany. Over to you.
Great, thank you, Rhonda, for that introduction. Hello, everyone. Thank you for joining our webinar today. My name is Tiffany Burchill. I am a technical support scientist here at ACD, and I will be sharing tips and tricks for one of ACD's newest product offerings, the miRNAscope assay, which was specifically designed to detect small non-coding RNAs, such as microRNAs, and short therapeutic oligos, such as small interfering RNAs and antisense oligos. On the agenda for today, I'll first introduce you to ACD's product portfolio with the use of a wide range of applications. Then we'll get into the details of the miRNAscope assay to detect smaller RNAs. I will then share some guidelines for preparing your samples, troubleshooting tips, along with some examples of the staining results. I'll then talk about the common applications that necessitate the use of the miRNA assay, along with some beautiful data to share with you. Finally, closing out with some frequently asked questions. To begin, I would like to talk about the importance of single-cell transcriptomics with spatial analysis. Previous attempts at detecting RNAs relied heavily on traditional bulk genomics analysis tools, such as qPCR or microarray. Although these methods deliver bulk expression levels, they do not provide detailed spatial information nor gene expression at subcellular level. Similarly, single-cell sequencing methods could offer a single-cell resolution, but still lack the important spatial resolution that researchers need to understand gene localization. So that brings us to ACD technology, where our advanced in situ hybridization technology enables single-cell spatial analysis of any gene from any species, allowing analysis of where exactly every single molecule is located relative to every other molecule in the tissue sample. The spatial and single-cell information is important for understanding pathological relevance of transcriptomes. The RNAscope technology is an ideal spatial analysis solution to interrogate complex tissues. It is both highly specific and sensitive, allowing for detection of RNA biomarkers in cells and tissues with morphological context at the single-cell resolution. An RNAscope technology consists of 3 parts: a unique target probe that ACD designs against your sequence of interest, a single amplification system that generates a high signal-to-noise ratio, and lastly, visualization of single RNA molecules as dots for a quantitative analysis. The assay allows for the spatial mapping of various RNA molecules, such as microRNAs, long non-coding RNAs, splice variants, point mutations, and highly homologous and short sequences in cells and intact tissues, all of which can be visualized with either fluorescent or chromogenic labels. In addition, the assays can be performed on a wide variety of sample types, including FFPE tissue, fresh frozen and fixed frozen tissues, and cultured cells. Next, focusing on the core RNAscope technology workflow, the RNAscope assay workflow is similar to immunohistochemistry, so researchers who are familiar with tissue-based protein assays find it quite intuitive to adopt this workflow. First, we start with properly permeabilized cells or tissues with ACD's pretreatment reagent. Then we hybridize the ACD probe to the target RNA of interest. Next, with the addition of the proprietary amplification system, the detection reagents allow the visualization of a single RNA molecule in the form of dots and enable us to quantify the level of gene expression indirectly. The combination of probe design strategy and the amplification system allows for high signal-to-noise detection of the target RNA. Our products cater to each of these steps by providing ready-to-use reagents in most cases. The highlight of the RNAscope technology is our probe design strategy. We can design probes for virtually any species and have done so in over 200 species, covering most applications highlighted on our website. The majority of our probes are designed for human and mouse. On our website, you'll be able to find the applications area to demonstrate how these probes are used in the most common applications, including infectious disease, cancer, neurosciences, and gene therapy. We've also added support for signaling pathways and indicated the probes we have available with that pathway. As of today, we have over 26,000 unique target probes in our catalog. Custom probes can also be designed against any target from any species in about 2 to 3 weeks. To design a specific probe for your target of interest, ACD requests you to provide us with either the accession number, information, or the exact sequence of the target. You can even indicate your region of interest for targeting with start and/or stop positions. Our bioinformatics scientists channel this information through the proprietary ACD alignment algorithm to select optimal sequence to ensure target specificity and uniformity of oligo hybridization in the assay. Our algorithm evaluates the sequence composition of each oligo in the pool to ensure no cross-hybridization with non-target RNA under stringent RNAscope conditions. Here we see the 3 unique assays that ACD offers to detect any RNA with spatial mapping at single-cell resolution. Starting from the left, RNAscope is designed to detect any mRNA or non-coding RNA that is greater than 300 nucleotides in length. RNAscope probes are designed in approximately 20 ZZ probe pairs, and the assays are available in either singleplex or duplex chromogenic, or as a multiplex fluorescent assay with up to 12 targets simultaneously in a single slide. The BaseScope assay, listed here in the middle, uses probes that are designed in 1 to 3 ZZ probe pairs to specifically support the short target detection of exon junctions, splice variants, point mutations, and highly homologous or short sequences between 50 and 300 nucleotides in length. BaseScope is available as a singleplex or duplex chromogenic assay. Last but not least, ACD offers the newest miRNAscope technology to detect the smallest RNAs and oligos, such as microRNAs, antisense oligos, siRNAs, and other small RNA sequences between 17 and 50 nucleotides in length. This assay is currently available as a singleplex chromogenic assay, which we'll be getting into more detail in subsequent slides. This table summarizes all the assays that are currently available from ACD. Our RNAscope assays are available as chromogenic singleplex brown and red assays, and duplex assays on both the manual and automated platforms. The RNAscope multiplex fluorescent assay allows for the detection of up to 4 targets simultaneously in a single tissue section. The major difference between the 2 fluorescent assays is that the V2 assay uses fluorophores from Akoya Biosciences with signal visualization and provides a high signal-to-noise ratio, whereas the fluorescent multiplex assay is an all-in-one kit. The V2 fluorescent assay is also available on the Leica automation platform. For higher plexing, we have the RNAscope high-plex assay, which enables the detection of up to 12 targets simultaneously in a single tissue, and is currently available in the manual format only. The BaseScope singleplex assay is available on all platforms, while the duplex assay is available on the manual platform. Finally, expanding our product portfolio, we now have the miRNAscope assay available as a chromogenic singleplex red assay. Overall, the miRNAscope assay is a robust in situ hybridization assay enabling highly sensitive and specific detection of small non-coding RNAs, such as microRNAs and short oligos, as ASOs and siRNAs in tissues with spatial and morphological context at single-cell resolution. In addition to these key applications that I just mentioned, the assay will also enable the detection of any small RNAs that are 17 to 50 nucleotides in length. Key research areas for these top targets include cancer research, infectious diseases, neuroscience, and therapeutic target validation. Additionally, this new assay leverages ACD's core amplification system and probe design capability to enable the detection of RNA molecules at single-cell resolution while improving high signal-to-noise detection of smaller RNAs with ease of data interpretation. To enable further insights into your research, the miRNAscope is also compatible with immunohistochemistry or immunofluorescence assays, allowing the detection of these small RNAs and proteins simultaneously on the same tissue slide. One of the key features of the miRNAscope assay is the probe design. The oligonucleotide-specific probe is designed using an informatics algorithm that selects sequences to specifically bind to the target sequence and not cross-hybridize with any other sequence. The bottom of the probe design complements and hybridizes to the target transcript, whereas the top of the probe is the base for the amplification structure upon which a preamplifier can bind, and the amplification tree can then be built. Once the preamplifiers are hybridized to the target probe, multiple amplifiers are then added to the preamplifiers. Each amplifier can then fortify multiple labeled probes, sequentially hybridizing to assemble a branching complex at each probing binding site. Labeled probes contain a chromogenic enzyme that generates a visible signal after the hybridization to alkaline phosphatase labeled probes and detected using a chromogenic substrate. Fast red is used as a detection reagent, which enables short RNA molecules to be visualized as red chromogenic dots under a bright field microscope. The entire amplification system allows for highly sensitive and specific visualization of target RNAs as punctate dots. Oops, went a little far there. Now we will focus on the overview of the miRNAscope assay workflow. The new miRNAscope assay workflow is very similar to the RNAscope singleplex workflow for those of you who are familiar with our technology. First, the cells or tissues that are bound to a slide are permeabilized using ACD's ready-to-use pretreatment reagents. The assay supports a variety of sample types, including FFPE, fresh and fixed frozen tissue, and cultured cells. Then, the small RNA target probes are hybridized to the target RNA for a single RNA target detection. The probe hybridization step is universal across all targets, meaning further probe optimization is not required. After probe hybridization, the target probes will be hybridized with ACD's modified branch signal amplification trees, consisting of amplifiers and labeled probes to provide high sensitivity and specificity. After the colors have been developed, the small RNA molecule signals can be seen as red chromogen deposits using bright field microscope. The slides can then be scanned for further quantitative and qualitative analysis. The entire singleplex assay can be done either manually or fully automated using the Leica system. Shown here are some recommended accessories for running the manual assay. At the top left are the two options for washing and handling slides. You may use either the TissueTech wash tray, common in most labs, or you can use our Easy Batch slide processing system, which was designed to streamline the assays and save handling time when working with a large number of samples. As the Easy Batch tray securely contains all slides that allow you to handle all 20 slides at once rather than each slide individually. The HiVIS hybridization system to the right is designed to maintain the required temperature and humidity to ensure optimal and consistent staining. In the bottom row are two methods for carrying out the heat-mediated target retrieval step required for some sample types, such as FFPE and fixed frozen. Our website has training videos on the different steps of the manual assay, which is a great visual guide to assist with running our assays. The standard miRNAscope kit configuration includes the necessary pretreatment reagents, the detection kit of choice, the wash buffer, and control and target probes needed to run the assay. This is an overview of the miRNAscope singleplex red chromogenic assay workflow using FFPE samples. As with RNAscope and BaseScope, the miRNAscope assay also has 4 major steps: pretreatment, hybridization, amplification, and finally staining and detection. The assay can take 8 to 10 hours and can be split between 2 days. Please refer to the user manual for sample preparation and pretreatment recommendations for your specific sample type. Pretreatment step is key in getting successful staining, so please make sure to follow the protocol exactly as described. I will highlight some of these specifics later in the presentation. Some general tips to ensure success with the miRNAscope assays are, first, to follow the protocol. We have various protocols with necessary details to help address the best way to run our assays. The miRNAscope assay user manual contains sample prep and pretreatment for most tissue types and the assay protocol. The sample pretreatment is very important to ensure that you get the optimal RNAscope signal. The pretreatment condition will differ from sample type to sample type and may require some optimization, so be sure to review the sample pretreatment recommendations for your sample type. Using controls is highly recommended to assess sample RNA quality and optimal permeabilization, and especially when you're troubleshooting your assay. And lastly, it is critical to make sure that you have all the required components before you begin the assay. Reviewing the checklist ensures following the protocol and reduces possible user error. On this checklist are some other key components that can help ensure your success with the miRNAscope assay. For example, using the HybEasy hybridization system, as I mentioned, this system has been designed to aid in handling of the slides. SuperFrost Plus slides are recommended to provide the best tissue adhesion to secure tissue sections, and EcoMount mounting slide medium to view the chromogenic signal. We also have recommended general guidelines, such as once you begin the assay, do not let the slides dry out. Warm probes and wash buffer at 40 degrees if any precipitation is present, and especially fresh reagents like 10% NBF or 12% formaldehyde, which is needed for the post-fix step. Right. Oops, I'm losing. In addition to previous guidelines, we also recommend running controls with your samples. The miRNAscope assay offers universal positive and negative control probes that will work for any species or sample. For the positive control, we recommend the small nuclear U6 probe, which is known to be expressed in most tissue samples. For example, on the top row here, shows strong nuclear U6 signal observed in a variety of tissues, including human and mouse tissues, and Hela and 3T3 cell pellets. A universal scramble probe shown in the bottom row is recommended as the negative control probe to demonstrate assay specificity and to be used for data interpretation against your target. The miRNAscope assay data could be analyzed with either SEMI-QUANTIFICATION or a single cell analysis with a software analysis tool such as Hela or other open source software analysis tools. The SEMI-QUANTIFICATION scoring criteria shown here is specific for the miRNAscope assay and provides guidelines for target expression levels as counted by dots per cell. As shown from the left, if your target is highly expressed, such as with 20 dots or more per cell, then we recommend a score of 3. Whereas if your target is moderately expressed with 11 to 20 dots per cell, a score of 2 is assigned. The lower target signal detected, such as 2 to 10 dots per cell, a score of 1 is assigned. Lastly, a score of 0 is assigned if there are no staining or less than 1 dot per cell observed. Please note that due to the relatively small size of the microRNA or ASOs or siRNA targets, the punctate dots seen under the microscope may represent multiple copies. As a result, we recommend our users to interpret the data by relative quantification, meaning that they should always use the scramble probe negative control as a baseline for measuring the change in the target probe expression. As I showed previously, it is important to review the sample-specific pretreatments recommendations, as well as sample preparation for optimal success with the miRNAscope assay. Here is an example of recommended sample preparation. With FFPE samples, we recommend fixing it in 10% NBF for 16 to 32 hours at room temperature. Once your samples are processed and embedded, the recommended sample thickness is 4 to 6 microns, and we also recommend that the tissue is mounted onto SuperFrost Plus slides. In order to preserve RNA quality, it is recommended to store sections with desiccants at room temperature for up to 3 months. For long-term storage, it is best to store at 4 degrees with desiccant. With proper sample prep and pretreatment, you can see robust assay performance in various species of formalin fixed paraffin embedded tissue samples. Here, miR124-3P is detected in the mouse, rat, and human brain tissue with abundant signal detected as expected. To demonstrate specificity, a known miR124 negative tissue, such as the mouse liver, was also stained with the miR124 probe. As you can see in the image on the right, the negative tissue shows no background detection of the miR124 target, so we can determine that we are seeing true signal with proper sample prep and pretreatment. A few highlights are present here for fresh frozen samples. Here we recommend storing frozen tissue blocks or sections in airtight containers at -80 degrees for up to 3 months. The recommended sample thickness is 10 to 20 microns, again mounted on SuperFrost Plus slides. The miRNAscope assay is also compatible with fixed frozen tissue and cultured cells. Similar detection robustness with fresh frozen tissue is demonstrated here. In the left column, miR124-3P is stained in the fresh frozen mouse brain tissue with abundant signals similar to what we saw with the FFPE tissue. Here the scramble probe is used for the negative control to demonstrate assay specificity with no background detected. In the right column is another example of fresh frozen tissue, but stained with miR-122.5P in the mouse liver, showing robust detection of the target, and no background is detected with the scramble negative control. If you are working with cultured adherent cells, the miRNAscope assay performs extremely well, as demonstrated in the slides with miR-205 stained in A431 skin cells. The assay successfully detected abundant miR-205 signal in the skin cells, and again, the scramble probe was used as a negative control, showing no background. Overall, the miRNAscope assay is a highly robust assay that can work with any of your sample types with proper sample prep and pre-treatment. Please contact tech support for sample preparation and pre-treatment guidelines with cultured adherent cells. Let us now review some troubleshooting examples and the results after performing optimization on these samples. miRNAscope uses universal assay conditions where minimal optimizations are required for successful detection of your target. With that said, when running the miRNAscope assay, here are a few factors that can affect the outcome of your results. For example, some problems are listed on the left, with fixation conditions are not optimal or hybridization conditions are not optimal. These factors and issues can be addressed by following solutions, such as the ones listed here on the right. Fixing samples in the recommended conditions for your sample type, as shown before in the FFPE tissue, and using the HYBES hybridization system for optimal hybridization conditions. One of the common issues that we see is related to tissue adherence to the slide. To troubleshoot sample detachment, we first recommend SuperFrost Plus slides to ensure optimal adherence. If you observe tissue detachment, the likely causes are listed here. We have found that including an additional baking step can mitigate this issue, as you can see in the before and after images. I will now review in detail the additional baking steps incorporated for FFPE and also fixed and fresh frozen samples. These additional baking steps can be applied to all assay types. For an FFPE sample, if you are observing detachment prior to the post-fixation, then adding a baking step before and or after the post-fixation, or if you are observing tissue coming off from the slides after the hydrogen peroxide or target retrieval, add an additional baking step before the protease step. All baking steps are for 30 minutes at 60 degrees in a circulating dry oven. In addition, ensure that you maintain a mild boiling of the target retrieval reagent to avoid bubbles getting trapped between the tissue and the slide. For sample detachment issues with fixed frozen tissue, we recommend drying the slides at -20 overnight after cryosectioning and adding a baking step after both post-fixations for 30 minutes at 60 degrees. We have found that the majority of the sample detachment happens after the target retrieval step, and so we recommend adding an additional baking step for 1 hour at 60 degrees before the protease step. After trying the steps 1 through 3, if you continue to observe tissue detachment, then we recommend that you extend the protease time to 45 minutes. For sample detachment issues with fresh frozen tissue, we recommend extending the post-fix duration at 1 hour at 4 degrees, and also including a baking step at 37 degrees for 30 minutes. If you continue to experience issues with detachment or other issues as mentioned earlier, please contact technical support for additional guidance. Now I will provide some detailed examples on how the miRNAscope assay could help with your research applications to enable detection at a single cell resolution for small RNA that can be synthetic or endogenous between 17 to 50 nucleotides in length. The short RNA nucleotides include antisense oligonucleotides, commonly referred to as ASOs, small interfering RNAs or siRNAs, and short hairpin RNAs or shRNAs. In addition, the miRNAscope assay can detect endogenous microRNAs between 18 to 24 nucleotides in length, and they have been shown to regulate various signaling pathways from development to adulthood. In the last few years, microRNAs have been identified as biomarkers in cancer and other conditions with other small RNAs like piRNAs and shRNAs that are between 26 to 34 nucleotides in length that silence transposable elements within the germline. miRNAscope is also fully compatible with immunohistochemistry, allowing the detection of both microRNA and protein markers. Some of the key applications for the miRNAscope assay are are in basic science in fields of neuroscience, stem cell research, and developmental biology. Infectious diseases in terms of inflammation and immune cell regulation, particularly important in viral infection. Biopharma therapeutics, specifically siRNAs and ASOs, have gained attention for their potential to be delivered in a tissue-specific manner. And cancer and clinical research where the small RNAs can be used to study tumor biology, immuno-oncology, and pathology. The miRNAscope assay can play a significant role in detecting microRNAs in a few key applications. In oncology, microRNAs have been shown to alter the transcriptional landscape within single cells, and in this way may induce malignant transformation of those cells by either repressing the expression of tumor suppressors or indirectly promoting the expression of oncogenes. In neuroscience, microRNAs are involved in pathogenesis of many neurological disorders, neuroinflammations, and immune regulation. For immunology, microRNAs have the potential to regulate immune cell activation, which is particularly important in viral infection and the immune system response. I'll now show some specific examples in the oncology research area. The markers shown here are known to play a role in these two types of cancer: cervical cancer in the top row and head and neck cancer in the bottom row. These markers are known to deregulate signaling networks that promote malignant transformation. For example, miR-205-5P, which is shown on the far left, this particular microRNA is known to promote tumor invasion, migration, angiogenesis, and one of its key targets is AKT activation, where the proliferation of cells are activated and initiation and progression of the tumor begins. This is similar to miR-21-5P, which also has been identified as a marker to protect the clinical outcome of cervical cancer patients. Both miR-155 and miR-233 have been widely studied in the context of the immune response to various conditions, including viral infections and cancer. miR-155 is an immune-enriched microRNA that is particularly expressed in cells of the lymphoid origin, for example, T cells and B cells, while miR-223-5P, also an immune-enriched microRNA, is commonly observed in cells of the myeloid origin. So cells such as macrophages and neutrophils tend to express miR-223-5P. As you can see, these two microRNAs are observed in these two cancers, suggesting infiltration of these key cell types, macrophages potentially and T cells, and they are observed at different expression levels. As a control, a scramble probe was used as a way to show that the staining we are seeing is true signal, shown on the far right. Looking now at neuroscience research, are other microRNAs that have been studied previously. In the left column, we have miR-124, which I mentioned earlier, but here we have a zoomed in image of the signal in the bottom row, so we can see that miR-124 is a very enriched microRNA in the mouse brain. Then we have LET7b5P, which is part of the well-known LET7 family of ultra-conserved microRNAs. LET7b has been shown to play numerous roles across all tissues. In this case, LET7b is involved in central nervous system pathologies, as well as tumor growth and immune response. We're seeing very strong staining of LET7b in the cerebellum. Next, we have miR-138-3P, which shows you the spatial resolution of this particular assay. It provides the morphological context to understand the function of this microRNA, as it is particularly expressed in Purkinje cells in the cerebellum. This microRNA plays an important role in cellular differentiation and maintenance of this particular cell type. Then we have a cortical region expressed miR-128-5P. This microRNA plays a significant role in neurogenesis and symmetrogenesis from very early stages in mouse brain development to neuromigration and outgrowth that we can visually observe in this particular section. Again, we show there is no signal with the scramble probe in sequential sections of the mouse brain on the far right. Another key application with miRNAscope assay is developmental biology. This particular image is a good example of the spatial resolution of the detection of miR-124. Here we have a sagittal section of a mouse embryo age E15.5. This image shows very nice detection of miR-124, specifically in the brain and central nervous system, but not in other regions. In comparison, we also show the scramble probe to show the robustness of miRNAscope assay. We can see very strikingly how miRNAscope delivers that spatial resolution that is required to fully understand the function of this microRNA. Another key application to discuss is small interfering RNAs or siRNAs. This microRNA has shown very significant potential in these key areas, which include oncology, neuroscience, and immunology. In terms of oncology, these siRNAs have shown to be a great therapeutic agent targeting key oncogenes that downregulate key signaling networks in different cancer or tumor growth. For neuroscience, there is research and clinical applications to use as a clinical treatment of acute brain injuries, as well as neurodegenerative disorders. With immunology, siRNAs can regulate or induce an immune response to a particular immune disorder, or even target particular genes that are deregulated in autoimmune disorders, and in this way, mitigating the disease progression. This is an example of a recent project that was completed through our assay services at ACD. This slide shows the specificity of the assay. In this project, 3 cohorts of animals were delivered either saline as a negative control animal on the left, another cohort with the siRNA, which we are calling siRNA1 for this presentation, and a third cohort of independent siRNA molecule called siRNA2. All 3 cohorts were tested with the siRNA1 probe, with only the detection in the tissue that we should be observing detection shown in the middle, with the other 2 tissues on each side of siRNA as negative. This type of actionable data can now be used to address other key things related to siRNA. For example, if there is any particular modification that would be beneficial, so you can examine biodistribution of siRNA as a function of different modifications. You can also examine the stability of siRNAs after increasing doses or titration of a particular siRNA. You can also study the impact of this siRNA as a function of the target that you are focusing on. There are multiple applications for miRNAscope in terms of siRNA. Another example of a key applications are ASOs. These are antisense oligos that are used in 3 key research areas, which include oncology, neuroscience, and immunology. In terms of oncology, ASOs are synthetically designed that target and block oncogene expression, which can slow down the growth of the cancer. In neuroscience, ASOs have emerged as an important therapeutic strategy to target genes that were considered untreatable with the drug therapy. Many of these targets are now moving into the clinical trial pipeline, and some ASOs have already gained FDA approval for treatment of a number of neuromuscular disorders. And finally, we have synthetic oligonucleotides that can target immune regulators and several diseases, including autoimmune disorders. Another project that we have completed through our assay services at ACD that shows the detection of the antisense oligonucleotide in 2 types of tissues, mouse cardiac and quadricep muscle. 2 different cohorts were delivered either vehicle or were treated with an ASO that they were trying to detect. As you can see, the ASO probe specifically detects the ASO treated animal as shown by the red chromogen signal, whereas no signal can be observed in the vehicle treated sample. This data allows you to formulate further hypotheses on how you can improve antisense oligo delivery by adding modifications conjugating the ASO to be delivered to a particular tissue of interest. You can also study biodistribution as it is expressed in a particular tissue, or if it is more systemically delivered into the different tissue within the cells, or it's within the animal, excuse me. Finally, we arrive at our last application, which is detection of piRNA. There are 2 applications. One includes epigenetics and the other one includes oncology. piRNAs are considered the largest class of small non-coding RNA molecules expressed in animal cells and are slightly bigger at 24 to 36 nucleotides in length. In terms of epigenetics, piRNAs are associated with Pb proteins and are safeguarding the germlines of animal cells by maintaining genome integrity and fertility for mobile DNA elements known as transposons. In terms of oncology, there are a number of piRNAs that are abnormally expressed in numerous cancer types. However, their function in cancer and the mechanisms by which they operate remain largely unknown. Here we show the detection of 3 independent piRNA molecules in mouse testes. We have PIR1, PIR3, and PIR131121 that have been published and validated, and they interact with PIR proteins. So we know these are piRNAs. These are also germline specific, so they're not expressed in any other tissue. The piRNA appears as a perinuclear punctate dot, and as you can see from this image, it is specifically detected, and there's spermatogonia, which is undifferentiated male germ cells that eventually give rise to the mature spermatozoa. As a negative control, we again showed the scramble probe, which shows no staining in the same area in the mouse testes. And to demonstrate further specificity of the probes and overall the miRNAscope assay, we also stained a mouse liver with the piRNA 131121. As I mentioned, these are germline specific piRNAs, and we actually see no signal from mouse liver, which is a somatic tissue, demonstrating the assay specificity and sensitivity at a single cell resolution. Before we move on to frequently asked questions, I just want to remind you that my colleagues are still available to help address your questions. So please continue asking your questions through the chat window while I go through some of the frequently asked questions. As displayed here, you can go ahead and read through them and see if any of your questions happen to be in here. A lot of these have been addressed, in this presentation, but there are, a little bit more specific questions that you can also review here. And here's another set of questions, specifically, replacing 4% PFA instead of 10% MBF, which you cannot unless it is notified in the protocol. Again, miRNAscope can be combined successfully with immunohistochemistry or immunofluorescence. And just to let you know, for more information on our products and the miRNAscope assay, please visit our website at acdbio.com. Under the support tab, we have a wealth of information, including a troubleshooting guides, user manuals, tech notes, training videos for the manual assays, as well as previously recorded webinars like this one that cover a wide range of different topics and applications. For those of you who are just getting started with ACD's assays, we offer a new user program where we provide scientific consultation with one of our expert technical support scientists to ensure that you're successful with the assay. In addition to our products, we also offer in-house assay services, as shown here. For RNAscope-ish assays, we can help with acquisition and qualification of tissue samples through our trusted chain of tissue providers. We also work with third-party CROs and can easily manage the embedding and sectioning of the samples in your studies. We perform the RNAscope, BaseScope, and miRNAscope assays, which are routinely provided through our service panel. Other options include quantitative assessment and compile this into a report, which includes our findings as well as the materials and methods of the study. So this concludes our webinar for today, a little earlier than full hour. I would like to thank everyone for attending today's webinar. We will continue to be online for a few more minutes. Please feel free to send in your questions to our panelists who are waiting to answer your questions via our chat window. Again, as a reminder, this webinar will be recorded and available on the website. In the event you were unable to get a question sent through the Q&A feature, please find the contact information for technical support on this slide. You can go ahead and email us at support.acd@bio-techne.com. We also have a phone line available, or you can also look up the resources, like I said, at our acdbio.com technical support. And thank you again.
Wonderful. Thank you very much, Tiffany. What an excellent presentation.